Hi everyone. In today's class, we will be studying about one important type of thermal processing. So we've already studied about thermal processing in the last class. So thermal processing is nothing but we use a time and a temperature combination to kill the pathogenic organism. So this one important thermal processing is called as pasteurization. So we've learned about pasteurization in many areas. So let's get into detail. All right, what is pasteurization? So pasteurization is nothing but a heat treatment process. So we know this is a thermal processing. So it is definitely a heat treatment process, which is performed to kill the harmful pathogens in food. We already know they use a time and a temperature combination in a um, thermal processing. So pasteurization is also a heat treatment where a time and a temperature combination is used to kill the microorganism. So the pasteurization uh, is the heating of every particle at least to 63 degrees Celsius and holding at such temperature at least for 30 minutes, or they will heat it up to 71.5 degrees Celsius and they would hold it for 15 seconds. Okay, what are the temperature and time combination here? It is nothing but either it is 63 degrees Celsius for 30 minutes, or it is 71.5 degrees Celsius for 15 seconds. Now, so we've learned like in thermal processing, there is always a time and a temperature combination. So this time and temperature combination always depends on the heat resistance. So heat resistance of what? Heat resistance, the particular vegetative or pathogenic organism that grows in a specific food product. Okay, I'll just repeat. This time and temperature combination depends on the heat resistance, but we should be very careful. This heat resistance is of the particular vegetative or pathogenic organism growing in a specific food. The second point is that it also depends on the sensitivity of the product quality to heat. So each product, okay, they have a different variation in the sensitivity to heat. So this sensitivity, okay, is also one of the important factors what time and temperature depends on. Now, let's get into detail about the objectives of pasteurization. So not only pasteurization, any kind of thermal processing, we always know why we do thermal processing, just to kill the harmful microorganisms. When harmful microorganisms are killed in a particular food, what happens? The shelf life can be increased, or the quality of the product can be increased, or what it helps in various consumer aspects. Okay, so here some particular objectives of pasteurization are the same as we discussed. It is nothing but it keeps the milk or any product safe for human consumption. Okay, that is a major criteria in any kind of process. Now, it also increases what? The keeping quality of milk. So when the keeping quality of the milk is increased, the storage, okay, can be increased, the shelf life can be increased. Thus, it pertains to what? The consumer's expectation. So this is one of the objectives of pasteurization. Now, alright, so when we have some kind of objectives or when we have some kind of advantages, we definitely have disadvantage, right? So when you talk about disadvantage, particularly for thermal processing and particularly in thermal processing, we're studying about pasteurization, right? So when a food is boiled or heated, what happens is that the nutritive value will always decrease. Okay, particularly in pasteurization, the nutritive value decreases. And there's something called as cream line. So what do you mean by cream line is that It is the amount of fat that rises, okay, that rises to the top of a bottle of milk. Okay, the amount of fat that rises to the top of the bottle of milk is called as a cream line. So this cream line is also affected. Okay, the cream line is also what? Affected. Next, clotting of milk. The clotting of milk usually happens with the rennet. Okay, that is a particular uh, enzyme, like it is called as RENNT. So the clotting of milk with rennet is also affected. Okay. Now, so when you subject any kind of product to any kind of processing, what happens? The cost also increases. Okay. Now, let's learn about the bacterial toxins. So what they say is that in pasteurization, when for example we take milk, when milk is boiled, uh, only the bacteria, okay, or the pathogenic organism is destroyed, whereas the bacterial toxins still remain in the milk. Now, so, okay, this is also one of the disadvantage. So, what do you mean by the inferior quality milk? That is nothing but if your milk, okay, or milk or any kind of product is inferior, or it does not meet the particular standard, but when it is processed, okay, what will happen is that it would mask the inferior quality of the particular product. Okay, it would mask the inferior quality of the particular product. Now, what are the disadvantages of pasteurization? A quick revision. The nutritive value decreases. The cream line gets affected. What is cream line? It is nothing but the amount of fat that rises to the top of the milk. Okay, when you just boil the milk, the fat rises, okay, to the top layer. So that is called as a cream line. So clotting of milk, especially the clotting of milk with a particular enzyme called the rennet, is also effective. So this cost also increases because when we process some food, there is a particular cost, and this pertains an increase in the cost of the product. Now, the bacterial toxins are not destroyed, okay? The bacterial toxins are not destroyed. Now, the last point is that it masks the inferior quality product, okay? It will mask the inferior quality product, not only milk, it masks any kind of inferior product. So we learned about the time temperature combination. We also know this time temperature combination usually depends on the heat resistance of the organism, or it also depends on what the sensitivity of the product quality to heat. Now here, okay, here particularly we take pasteurization in pasteurization process for milk. Uh, this time temperature combination usually depends on the thermal dead point of pathogenic organisms. Okay, they say that the temperature time for pasteurization process is principally based on the thermal dead point of the pathogenic organism particularly. So what is the principle? Okay, principle behind the temperature and time combination is that they usually they usually depend on they usually see to the thermal dead point of pathogenic organism. The other thing is that the Mycobacterium tuberculosis. It is one of the most heat resistant pathogenic organism. So this should also be destroyed. So now the other thing is that the phosphatase enzyme. Okay, now we are talking particularly about the milk. So, okay, we'll have this Mycobacterium tuberculosis or phosphatase enzyme. So what they say is that the temperature required to kill Mycobacterium tuberculosis is much lesser than the temperature required to destroy this phosphatase enzyme. So they usually go for a time and a temperature combination that kills both, okay, that kills Mycobacterium tuberculosis and also destroys the phosphatase enzyme. Okay, very, very important. Okay, that is nothing but thermal dead point of the pathogenic organism. So pathogenic organism in milk is Mycobacterium tuberculosis. So you have to choose a time and a temperature combination that kills the Mycobacterium tuberculosis and also the phosphatase enzyme. Now, so in pasteurization, there are different processes. Okay, we'll get into detail, before that we should be knowing certain process like low temperature, okay, low temperature short time process, high temperature short time process, or ultra high temperature. So these are the important, okay, important. They might ask you in any exams. If suppose, okay, you have a pasteurization topic, people usually ask about the time and the temperature combination. Be very careful about it. If it is for 63 degrees Celsius, it is 30 minutes. If it is for 72 degrees Celsius, it is nothing but 15 seconds. And in ultra high temperature, just remember, okay, if we are going to heat a food product to 100 degrees Celsius, the holding time would be 0.001 seconds. Okay, just okay, note down the highlighted points. They are very important. Okay, now, so they say that at such a particular time and a temperature combination, okay, when you combine such a time, I mean a temperature and the time, the Mycobacterium tuberculosis and also the phosphatase enzyme, uh, they are both are destroyed. Okay, please learn the time and the temperature combination. Now, what are the methods of pasteurization? We've got like in-bottle pasteurization, batch pasteurization, HTST. HTST is nothing but high temperature short time pasteurization, vacuum pasteurization, flash pasteurization, and ultra high temperature. Now, in-bottle pasteurization. In-bottle pasteurizations are nothing but, if you see the picture, we'll have a clear-cut idea of what is in-bottle pasteurization. So, for example, when raw milk, mostly raw milk, okay, are filled in glass milk bottles, so they will be tightly sealed with special caps. So these bottles will be subjected to heat treatment. Okay, heat treatment. When it is a heat treatment, always remember there is a time and a temperature combination. For in-bottle pasteurization, the time and the temperature combination is 63 or to 66 degrees Celsius for 30 minutes. So after a heat treatment, there will always be a cooling process. This cooling process, okay, uh, okay, will bring down the temperature of the produce or the product to 4 to 5 degree Celsius. So this is only in bottle pasteurization. Nothing but milk filled in a glass bottle will be subjected to heat treatment. So before subjecting to heat treatment, the glass bottle should be tightly sealed. Okay. After it is being tightly sealed, it has to be subjected to a heat treatment or of combination 63 degrees Celsius for 30 minutes. After the heat treatment, the bottle, okay, or the milk bottle should be cooled to 4 to 5 degree Celsius. Now, I suppose you're clear with the in-bottle pasteurization. So, what are the demerits of in-bottle pasteurization? So, when we talk about the demerits, so when you subject a glass bottle to such a temperature, there are chances for bottle breakage. Okay, there are chances for what? Bottle breakage. Next, they say that, okay, the heat transfer during heating as well as cooling, okay, is very low. Okay, the heat transfer is what is very low during heating as well as cooling. Now, for this in bottle pasteurization, special type of machines and caps are required. So when special types of machines or caps are required, what happens? The cost of the product also increases. Now, there's one concept that when you heat any kind of product, like okay, there can be an expansion. Okay, there can be an expansion of the product. So in order to meet this, we require oversized bottles. Okay, we usually require oversized bottle. So this is also one kind of a demerit. Okay, this is also one kind of a demerit. Okay, now next, batch pasteurization. Okay, you have an important MCQ here. So batch pasteurization is also called as the VAT pasteurization or Just note it is LTLT, low temperature, long time pasteurization. Here it is similar to in-bottle pasteurization. The time temperature combination is 63 degrees Celsius for 30 minutes, and also it is cooled to 4 to 5 degree Celsius. Okay. So this batch pasteurization usually happens, okay, in a vat pasteurizer. So this vat pasteurizer consists of what? An agitator, okay. That is why it is called as batch pasteurization, but because it is done in vat pasteurizer and it contains, okay, this pasteurizer would contain an agitator and a thermometer, okay, agitator and a thermometer. Okay. Now, so talking about the advantages and disadvantages of batch pasteurization, okay, it is suitable for small operation, okay, it is actually suitable for small operation, and the process is what? Very simple, and no milk is lost during this pasteurization process. But whereas when you get to the disadvantage, as we all know, it is a long time process. The time taken for this process is what? Is very high. And we use a vat pasteurizer, and the cleaning process is also very tedious. Okay, so that's all with the disadvantage of batch pasteurization. Yeah. Now, high temperature, short time pasteurization. So this pasteurization usually requires a temperature of 71.5 degrees Celsius. So 71.5 degrees Celsius means that what um, the holding period, okay, will be very less, okay, hardly some 15 seconds, okay, holding period is very less, like 15 seconds, or very or seriously very less holding period. Now, so this HTSD is also called as a continuous process of plate pasteurization. Now, vacuum pasteurization. Vacuum pasteurization is also called as vacuation. So what is the use of this vacuum pasteurization is that it removes of flavor, okay, very important. That type of pasteurization removes of flavor is nothing but vacuum pasteurization, or vacuum pasteurization is also called as vacuation. So what is the principle of this vacuum pasteurization is that when you, okay, here milk is mixed with pressure steam, and this immediately followed by a sudden and a spontaneous expansion, okay, into a finely divided state with the chamber maintained at reduced pressure. What they say is that this milk, okay, M stands for milk, and milk plus what? They go with a pressure steam, PS. Okay, I'm denoting it as PS. So milk when pressure steam. This is immediately followed by what? A, um, a sudden and a spontaneous expansion. So this is what the principle behind vacuum pasteurization. Now, next topic, flash pasteurization. Flash pasteurization is similar to HDSD. Here, the temperature is 80 degrees Celsius and there is no holding period. And the biggest advantage is that the reduction in nutritive value is very less. And the time taken is also very less. Okay. And this process destroys all the pathogen organisms that might be present in milk, and it also makes it safe for human consumption. Okay, now UHT, the last process, ultra high temperature pasteurization. So here the temperature would be 135 to 150 degree Celsius. So what is the important advantage? Okay, advantage here is that it maintains the nutritive value. Okay, here the nutritive value is maintained, and the color, flavor, okay, is maintained, similar to that of a raw milk. Okay, color, flavor is also maintained and stands for nutritive value, color, flavor is also maintained in UHT process. Okay, so that's all with pasteurization. I'll just give you a quick brush up of pasteurization. So pasteurization is nothing but it is a thermal processing that uses a time and temperature combination to kill the microorganism. So what are the different types of pasteurization? They are nothing but in-bottle pasteurization, batch pasteurization, high temperature short time pasteurization, vacuum pasteurization, flash pasteurization, ultra high temperature pasteurization. Here we need to be very careful about the temperature of each process. Okay, please jot down the important temperature and time combination in this lesson for your exam preparation. And that's all for today's class. Have a nice day and thank you for watching.